Hi everyone, I'm Srijay. I'm going to present the poster titled SPADESegResNet, Harnessing Spatially Adaptive Normalization for Breast Cancer Semantic Segmentation. The problem here we are targeting is the semantic segmentation, where we try to map each pixel from this input image region into the one of these classes. These include tumors, trauma, inflammatory, necrosis, and other tissue types. The task is challenging and essential for problems like cancer classification, tumor segmentation, and they offer valuable insights into diagnostics, prognostications, tumor response, and cancer biology research. There are a lot of existing deep neural networks employed for this semantic segmentation task, but they often lose key image details as we progress through the deep layers, and that's why limiting the performance. So to address this challenge and to retain the key image details till the last layer, we propose SPADESegResNet, which uses specially adaptive normalization layer, abbreviated as SPADE, for improved breast cancer semantic segmentation. Here is the detailed architecture of the framework. The idea of SPADE layer is taken from the SPADE image-to-image translation framework, where we try to generate image condition on some other input image, such as segmentation map. This is yellow colored 768 by 768 pixel region. This is passed through the convolution layer, which batch normalization ReLU activations, creating the latent representation, which is then passed through the spade race blocks, which include spade and convolution layers. In the spade layer particularly, input image generates modulation parameter, which adjusts the normalization activation. The output of spade race unit undergo transpose convolution, batch normalization ReLU activation, to upsample the image from the deep representation, creating the segmentation map of the same size as that of input. So basically, we slide through the regions of whole slide image, correspondingly generating semantic segmentation maps. Of course, there will be some overlaps, but the final input, the final segmentation map is generated by averaging the class probabilities of overlap regions. The dataset we use for these experiments is the BCSS breast cancer semantic segmentation datasets, where we extract around 5,000 images of 768 by 768 pixels and 20 for training and 2,500 images around for testing from 151 whole slide images taken from 151 patients. The results on the test dataset can be seen here and as well as here. The segmentation map predicted by SPADESegResNet framework, we can see that it matches closely with the original segmentation map. In these three examples, a careful examination highlights the exact segmentation achieved for tumor regions, shown in yellow color by our model, in contrast to UNIT and UNIT++. Additionally, our model effectively captures trauma, shown in green, and other tissue types in purple color, and demonstrate fewer instances of misclassifying necrosis, shown in dark blue color. To evaluate a model quantitatively, we employ three performance metrics: DICE index score, AUC, and the overall classification accuracy. The results shown in a table and also in box plots here highlight moderately superior performance of SPADESegResNet framework compared to other baseline models, particularly in the context of tumor and necrosis. We can also observe that UNIT++ exhibit notable accuracy in identifying trauma, but with comparatively lower metrics in other aspects. In terms of overall accuracy, SPADESegResNet achieves the highest accuracy of 77%. So to conclude, we showed that SPADESegResNet achieved moderately improved segmentation performance across various tissue types compared to baseline models such as UNIT and UNIT++. And this study opens various avenues for development of interpretable models for semantic segmentation task. Thank you.